My name is Josh Harrison, and I'll describe briefly our progress towards achieving the goals of Thrust 1, where we seek to characterize microbial biogeography within Wyoming and test theoretical predictions of microbial distributions using the data that we collect. So I'm a postdoctoral researcher on the project, and I work in Dr. Alex Berkeley's lab, and I was hired at the beginning of 2018. Broadly speaking, I'm an evolutionary ecologist, and I prefer to work on organisms living in and on plant leaves. And so I study the ecology of the phylosphere. So I mostly work with insects and plant-associated microbes. So there are three goals of Thrust 1. We've made progress towards achieving each of these goals as a research team. The three goals are to resolve the distribution of microbial taxa with spatial and temporal sampling, to define the distribution of functional traits of those taxa, and then to improve statistical modeling techniques. So in this brief presentation, I and my colleagues will give you some highlights of our progress towards achieving each of these three goals. So first, we've successfully collected samples from numerous locations from throughout the state of Wyoming, as you can see on this map. And so Wyoming is the lightly outlined rectangle that's in the center of the screen. And the blue stars represent sampling locations that were assayed in 2018, and then the red pluses are locations that were sampled in 2019. So a number of researchers were involved in sampling, and we've collected a large variety of substrates. So we've collected everything from plant samples to soil samples to water to sediment from the bottoms of lakes, snow and glacier melt, even air samples. And as you can see, we've sampled across a variety of abiotic and biotic gradients. We've done a particularly thorough job of sampling elevational gradients from throughout the state. So Wyoming is a geologically and biologically diverse state. We've got remote mountain ranges, beautiful deserts, wide expanses of high prairie, and additionally there are a lot of managed landscapes throughout the state, particularly rangelands and croplands. And so we've sampled from all these different habitat types. And so the scale of sampling that we've achieved so far will allow us to make substantial progress towards characterizing microbial biogeography across the state, and allow us to answer many basic questions such as how microbial diversity shifts with elevation, aridity, and as influenced by specific management regimes. So while time constraints preclude me from providing an overview of all the different ongoing projects, I'll describe a little bit of my own work involving phylosphere microbiota as an exemplar of the progress we've made toward this first goal of Thrust 1. And so by phylosphere, I'm referring to the above-ground tissues of plants, so leaves and above-ground stems. So some estimates suggest that the phylosphere has about twice as much surface area as the Earth. And of course, it's where photosynthesis happens. And so it's fundamental to ecosystem processes and life on our planet. And importantly, the phylosphere is absolutely filled with microbes. There's microbes that live inside of plant leaves, and those are called endophytes, and microbes that live on the surface of plant leaves, and those are called epiphytes. And we know that they likely have important roles to play in nature, but we don't know much about their biogeography, individual ecologies, and natural history. So, using the resources of the EPSCoR program, I resolved to address some of the basic questions regarding phylosphere microbiota biogeography. So for instance, I'm curious how endophyte assemblages might shift with elevation. What are the associations of different plant traits with specific endophyte taxa? How do spatial distributions of specific taxa shift across the state? So for instance, bacteria versus fungi, and so on. And so I sampled elevational gradients throughout the state, and these can be seen with these yellow dots with the black circle in the center of them. And so you can see they're scattered throughout the mountain ranges throughout the state. And there's also these blue dots shown here on the map, and these are sagebrush samples. And so sagebrush communities were sampled by my colleagues in the Microbial Ecology Collaborative, and they sampled both foliar tissue and soil samples. And so I represent these here because we'll share data between our two projects to improve our scope of inference and better address the goals of Thrust 1. And so the sagebrush samples are mostly collected in the desert and the high prairie, whereas the sampling that I did was primarily in forested regions and in mountains. So in sum, I sampled tissue from about 68 host taxa and 20 locations from across the state. So at each of these locations, I laid a 50 by 50 meter square and sampled 40 to 70 different hosts within that square, depending on the location. In total, I collected about 2,500 samples, and these include both epiphyte and endophyte samples. And I'm currently sequencing them for bacteria and fungi. So about total of about 5,000 samples to be sequenced, counting those two taxa. And so as the data roll in, I'll be able to link plant trait variation and then the abiotic characteristics of the site to variation in those epiphyte and endophyte assemblages, and hopefully put some of these long-standing questions regarding phytosphere biogeography to rest. And so with that, I'd like to turn it over to my colleague, Dr. Pauly as he describes our progress toward the second goal of Thrust 1. Thanks.
So for goal 1B, we just define the distribution of functional traits. We performed a couple of studies to look at this exactly. The first one was looking at spatial autocorrelation in microbial communities. This study was led by Gordon Costa, who's a PhD student in the Wyoming EPSCoR Microbial Ecology Collaborative. And the purpose for this study was to examine the effect of space on microbial communities. And to do this, we used a laser to pinpoint locations within the site to collect soil samples from, and this interval between sites ranged from 1 millimeter to 1 meter apart. And the idea was to examine the effect of space on autocorrelation in microbial communities collected from the same site. For this purpose, we were joined by Mr. Mark McCarthy, who is a K-12 science teacher in the alternative school in Casper, Wyoming. The second study was looking at microbiomes in hypersaline and inland freshwater ecosystems in Wyoming. This was led by me, Paul Ayayi. I'm a postdoc in the EPSCoR Microbial Ecology Collaborative here at the University of Wyoming in collaboration with Lasha Tronstad and Linda Van Depen. And we're interested in examining the differences between host-associated and free-living microbiomes within hypersaline and non-saline freshwater aquatic systems in Wyoming. And for the host-associated microbiomes, we're interested in investigating whether taxonomy of the microinvertebrate or the ecology of the microinvertebrate impacted host-associated microbiomes. So to do this, we collected samples from the Powder River Basin in Wyoming. The streams located in the Powder River Basin in Wyoming are characterized by being really saline compared to other freshwater systems in Wyoming and in general. And this salinity differs significantly from this other ones because of the inherent geomorphic nature of the rocks in the basin. Within this basin, we had 4 main sites, sites 1, 2, 3, and 4, from which we sampled water and biofilm samples as well as microinvertebrate samples. And we had a within basin reference, which was the South Fork River. This site has salinity relative to other inland freshwater systems, but it still has a really high salinity, still really relatively low salinity compared to sites 1, 2, 3, and 4. So for site 1, as an example, this is site 1, and the white layer you see here are the salt deposit on top of the water body, reflecting how saline it is. Whereas for site 3, even though it's relatively saline and hypersaline system as well, it is not as saline as site 1, and therefore lacks the white scum formation on top of it. We also then picked a non-basin reference site, which was the Laramie River, where we collected water and biofilm samples. So to summarize, we had 4 sites within the basin that were very saline. We had a within basin or basin reference site that was saline as well, but not as saline compared to the fall site, and a non-basin reference site, which is more typical of what you expect of an inland freshwater system. Some of the differences in terms of the abiotic characteristics of this site are shown over here. For SPC, which is specific conductivity, which is a measure of the saline or salinity of the streams. ORP, which is the oxidation reduction potential. That again is influenced by the salinity and the pH of the site. As you can see, site 1, 2, 3, and 4 are in order in terms of decreasing SPC as well as decreasing ORP. And the non-basin reference site, which is the Lyman River, has lower salinity, lower ORP relative to the basin reference stream, which was the South Fork River. Although again, it is saline, it is less saline compared to the 4 main site sample. Insert over here are the summaries of the salinity, SPC, and the ORP of all saline streams relative to the basin reference and the non-basin reference. We also collected a variety of macroinvertebrate taxa from the 4 streams, the 4 hypersaline streams, and the collected macroinvertebrate taxa were then grouped together into functional feeding groups. And there were 4 main functional feeding groups that we determined from the sample macroinvertebrate taxa. These were the predators, which are made up of different species of diasticity, and then included also the lastity. The garthreds, which are made up of 2 different species of hydrophility, 1 species of hydrinity, Chironomidae and Amphipoda, and then 1 mayfly species, Paracloides, which in the family Betae is a scraper, and then 1 species of hydrophility, which is a shredder. After sequence analysis, one main trend that stood out was that among biofilm samples, and then the water or the bacterioplankton samples, there tend to be significantly more taxa detected in the biofilm samples relative to the biofilm or the bacterioplankton samples. 2, among the functional feeding groups, there was no significant difference in taxa, but the general trend is relatively low taxa detected in the scrapers, although there's a high variability around that point relative to the garthreds, the predators, and the scrapers. In terms of community composition, among the sample, the various sample categories, 2 main trends again stand out. For the water or the bacterioplankton samples over here, which are the field colored markers, diamonds, the water sample tended to cluster separately from the biofilm samples. And 2, among the biofilm samples, the basin reference, the non-basin reference samples, which was the Lyman River biofilm sample, tended to cluster separately from the basin reference and then the saline biofilm samples. The third point is that the macroinvertebrate taxa collected seem to group together with the biofilm samples relative to the water samples. And even among these macroinvertebrate taxa, the differences among the garthreds, the predators, and the scrapers. And importantly, the shredders tended to cluster separately away from those 2, separate away from everything else over here, tend to be more similar with the biofilm sample from the non-basin river. The next couple of slides will then be led by Dr. Alex Berkeley, who will discuss the progress for Goal 1C related to advances in statistical modeling.
Hi there, my name is Alex Berkeley. I'm one of the co-PIs on the Microbial Ecology Collaborative Project, and I'm here to tell you a little bit today about Goal 1C as part of Thrust 1, where we are developing statistical methods, and in particular, we're coupling advances in statistical methods with our advances in laboratory methods. In particular, we are interested in using sequence data to characterize the composition of taxa in our samples. Often we're going to be able to have replicates or sampling along a gradient, which allows us to combine information from multiple samples and develop models that are appropriate for that combination. In our sampling, we're doing high levels of multiplexing, which results in unequal sampling effort amongst our different samples. And we want to have appropriate statistical models to deal with that, that variance in sampling effort. In another video, I'll tell you about some of our laboratory protocols for DNA analysis and amplicon analysis. But briefly, we're multiplexing up to 9,200 samples in a library, often for 2 loci within one library. But we have methods for 16S, for ITS, for CO1, and for loci used to detect the coronavirus. In our advances, we've incorporated means to incorporate an internal synthetic reference molecule and have developed methods for liquid handling that make our methods highly repeatable. So I'll talk about the laboratory protocols in a facilities video. But today in this short video, I'm going to focus on the statistical methods and some considerations to be able to move from relative abundances to absolute abundances. So what we're conceiving of and where we started was where we might want to be able to compare the true composition between a treatment and a control, which I visualized here on the left side with a composition with a set of taxa represented by different colors in the left horizontal bar, and another composition on the right in another horizontal bar, where the question is which of the taxa differ between treatment and control, and by how much. And so at the top level of our hierarchy, we have the individual X's, which are our counts of sequence reads that correspond to different taxa. Each one of those counts is a realization, a stochastic draw, because we have a finite sample from a true community. So it's a draw from a multinomial distribution with a true vector P that characterizes the frequency of all the taxa. But each one of those 5 replicates within treatment or control is itself a random draw or a random realization from an underlying distribution with a true frequency for characteristic frequency for that taxon in the treatment or the control that I've represented with a pi vector. That pi vector characterizes the expected frequency in treatment or control, and the theta parameter characterizes an intensity parameter that affects the variance for sampling from a Dirichlet. So the way in which we sample in microbial ecology fits naturally into this hierarchy, and we specified a model for this, where we would simulate data with known distributions, generate counts, and then in panel D, compare the frequency that we infer and do differential abundance testing, where we learn the extent to which individual taxa differed from one another between treatment and control when we expected them to. We competed our Dirichlet multinomial model against various commonly used alternatives, and we had a paper come out recently in which we were able to show that the Dirichlet multinomial approach outcompetes commonly used methods for differential abundance testing for microbial abundances or for transcript abundances in gene expression studies. So beyond the multinomial Dirichlet, we're also involved in setting up regression models to test things along gradients. We're not going to talk much more about that just now. You'll see an example of that in one of our other talks. But what I wanted to mention briefly is we have another more recent paper where we came to appreciate how much of a challenge our sampling was going to represent for comparing taxa across truly different types of samples, soil, water, and phytosphere, unless we had some type of internal standard that allowed us to resolve the causes of some of the frequency differences. And so what we've portrayed here on the top is that without an internal standard in the top row, suppose we have a composition where the relative abundance is 50-50 of the orange and the turquoise taxon, and we have a second sample in which the relative abundance is 0.7 and 0.3. There's considerable ambiguity as to whether C, D, E, or F is the true abundance that gave rise to that difference in relative abundance, because all of those bars to the right correspond to a relative frequency of 0.7 and 0.3. By adding in an internal standard into our composition, where we add a known molarity, and there are lots of considerations of how that should be done that we go into in the paper, there's the potential to be able to resolve whether that shift from 0.7 to 0.25 for the relative abundances of the taxon that are there correspond to different forms of true absolute abundances. By properly utilizing an internal standard, we have the potential to calculate absolute abundances from compositional data, but there are constraints on when that will be possible, and we've explored those. Overall, for Thrust 1, we're in really good shape in terms of making progress towards sampling broadly within the state and along different geographic gradients to learn about the distribution of taxa with spatial and temporal sampling. There are a few places that we noted in the annual report where we're a bit behind on learning about the distribution of functional traits, but we're making good progress in that area. And then finally, with respect to Goal 1C, we're in good shape in terms of making progress with statistical models and developing our statistical models in a way that directly fits with the laboratory procedures that we're doing. Thanks much.
Hello, I'm Linda Van Diepen. I'm a co-PI on the Microbial Ecology Collaborative. I'm going to be talking about our progress for Thrust 2, in which we measure microbial life and its functional consequences. Thrust 2 has two main goals. The first goal looks at the consequences of microbes in terms of how they affect biogeochemical cycling, and the second goal looks at how they affect organismal performance. For both goals, we're currently on track based on our strategic plan for year 3. As an example for this first goal, I'm going to talk about our sagebrush project. This project was a large undertaking led by a postdoc in my lab, Paul Ayaye, together with a postdoc in Dave Williams' lab, Felix Boudoir, and also several undergraduate students, of which Martina Greenhaw significantly contributed to both the fieldwork as well as sample analysis that is still ongoing. Sagebrush, and particularly big sagebrush, is the dominant species in mid-latitude shrub steppe ecosystems, which in Wyoming occupies about 1/3 of the state and can be found in at least 10 basins across Wyoming. These 10 basins together represent a climate gradient across Wyoming, which vary in both mean annual temperature and mean annual precipitation. And the 50 sites that were sampled last summer have some sites which are hot and dry, as well as some that are cooler and also wetter. At each of the 50 sites, the group collected both vegetation and soil samples. Each site consisted out of 4 plots in which a large primary and small secondary sagebrush plant were sampled to measure their leaf-associated microbiome. In addition, the top 5 centimeters of the soil beneath their canopy was sampled. For the secondary sagebrush plant, we also collected the root samples to study the rhizosphere microbiome. Lastly, we collected intercanopy soil, the areas between the sagebrush plant, to represent the influence of grasses and forbs on the soil environment in this climate gradient. This represented 3 different types of soil samples, as well as 2 types of plant samples to be analyzed for the microbiomes, as well as additional variables that are being measured for both soil and plant samples, including their carbon, nitrogen, and sulfur content, and isotopic concentrations, as well as other soil parameters such as pH, texture, and microbial enzymatic activities. Some of the preliminary results show that soil moisture varies significantly among the basins that we sample, as well as the different soil types, with in general lower soil moisture in the intercanopy bulk soil, which in general are less protected by vegetation and more exposed to the elements. Looking at the relationship between measured soil moisture and recorded mean annual precipitation across the 50 sites showed a weak but expected positive correlation, indicating that we were able to capture the climate gradient in our soil sampling to some degree, though some of the higher precipitation sites were apparently sampled during a much drier period. Looking at the relationship between some soil abiotic variables and mean annual precipitation, we see that there was no relationship with electrical conductivity, which represents soil salinity. However, soil pH had an expected negative correlation with mean annual precipitation, with decreasing pH as precipitation increases. And this negative relationship is caused by both the leaching of cations as well as increased microbial activity, such as the decomposition of organic materials. To further understand drivers of biogeochemical cycling across the sagebrush gradient, we're using some modeling. So far, based on the preliminary data we've collected and analyzed, we can see that soil-water dynamics are probably an important driver of biogeochemical processes in these sagebrush ecosystems, as both soil nitrogen content as well as delta 15N are correlated to mean annual precipitation, as well as both carbon and nitrogen content are correlated with soil texture, which determines the fate of the water. So whether it's drainage with high sand content versus storage with a higher silt and clay content. All of the isotopic measurements show strong correlation with soil pH, which can be a significant driver of microbial communities. In summary, the acquisition of these elemental results for all sites for both soil and vegetation, combined with the characterization of the microbiome, should really help to understand how environmental parameters control microbes and how that feeds back to biogeochemical cycling. We expect that this project will lead us towards the development of experiments that target pertinent microbial-mediated processes. For goal 2B, Cynthia Weinig is now going to present some of the experiments in her lab that is being conducted to understand plant-microbe interactions, with one of her postdocs, Riley Dibner, presenting some of her work on the effects of rhizosphere microbiomes on plant host plots.
My name is Cynthia Weinig. I'm a co-PI on the current project, and I'm going to talk generally today about plant microbe interactions and more specifically about the potential for local adaptation of plants to rhizosphere microbiomes. So plants and rhizosphere microbes affect each other through complex feedback loops. Plants have the possibility of affecting the assembly of microbial communities in part through exudation of different compounds into the soil, immune signaling, and also interactions and functions of the circadian clock. Microbes that are found in the soil also have potential effects on plant performance, including defense against herbivores and pathogens, access to nutrients, and tolerance of both abiotic and biotic stress. So one can imagine in this context that microbes could have diverse types of effects on plant performance. In the context of the current award, we've been looking at a range of model crop and native plant systems, depending on the question we're interested in addressing. For instance, we've worked in Arabidopsis thaliana, a genetic model plant, to examine the relationship between the host circadian clock and the rhizosphere microbiome. And this work was performed by Charlie Hubbard and Ali Soretto, who are graduate students on the project. In addition, we've worked in the crop Brassica rapa to look at feedbacks between host secondary metabolites and the rhizosphere microbiome. This work was done by Ella DeWolf, who's also a graduate student on the project. We've worked in Bucra stricta, which is a native Brassicaceae to Wyoming, to look at the effects of fire history and season on the microbiome and microbiome effects on the host clock. And that work was done by postdoc Riley Dibner. And additional work in Bucra stricta looked at plant local adaptation, and that work was done by Rob McMinn. We've looked at Nuphar polysepala, which is the montane water lily, and its role as a potential ecosystem engineer for aquatic montane microbiomes. And finally, we've been looking at how atmospheric deposition may vary as a consequence of vegetation assemblages that act as environmental filters and affect microbiome establishment. And that work was done by the undergraduate student Ida Lewis. So these are a range of different projects that we've been working on related to plant microbe interactions. What I'm going to focus on is the work done by Rob McMinn on plant local adaptation. And in the next talk, you'll be hearing from Riley Dibner about fire history and its effects on the rhizosphere microbiome. So if we consider local adaptation, this occurs when genotypes are better adapted to their local or home environment than are conspecific genotypes from foreign or so-called away locations under the model or construct of a home versus away advantage. Soil microbiomes may act to increase performance of all genotypes and fitness, and this is illustrated in the top panel here, where we see that host genotypes A and B, so these are the plant genotypes A and B, shown in green and orange, differ in their performance depending on whether they're grown in the presence of, shown here on the right, or in the absence of a soil microbiome. So growing with a microbiome improves plant performance. Further, local genotypes may be adapted to their home microbiomes and display a differential fitness benefit. That is, local adaptation may occur. We can see this illustrated in the lower panel, where host genotype A, shown in green, has a performance advantage over host genotype B when grown in the presence of its home or local microbiome. The reverse also is observed, where we see that host plant genotype B has a performance advantage over genotype A when grown in the presence of its own local microbiome. When growing plants in controlled settings, we found that there was local adaptation. So we'd collected seeds from over 40 local populations of Bucra stricta. This is shown across the map here on the top, several different mountain ranges, and the circle icons represent the populations that were collected. Soil from these local populations was used to inoculate plants that were raised in controlled greenhouse settings, and growth was then measured. And in the lower panel here, we can see illustrated that some soil microbiomes improve the growth of local plant genotypes. Across the bottom here of this panel is the soil microbiome from three different sites plus a disrupted or controlled treatment. We measured total biomass, and the lines here show the genotypes that were raised in these soils. And what one wants to notice is that South Brush Creek genotype performed better than the other two genotypes when grown in the presence of its home microbiome. Likewise, Webb Springs genotypes performed best when grown in the presence of the Webb Springs microbiome. The Coal Creek site has been found in multiple studies that we've performed to be pathogenic. That is, it decreases plant growth. And so that's not surprising. There's no evidence of local adaptation to that soil microbiome. But these patterns where there's a reversal in the performance across the South Brush Creek and Webb Springs soils suggest local adaptation. We find the same results effectively when we used a field experiment. So in a reciprocal transplant of genotypes across either an intact home site at Crow Creek, another site we refer to as Road 234, or a third site, Webb Springs, we find that overall, the presence of having a soil microbiome in those sites does improve growth on average over all genotypes. But note again that Road 234 has a significant advantage in terms of plants that survive there relative to the two other genotypes grown in the Road 234 soil. And the same is true at another site. If we look at the lower panel here, we find that the Webb Springs genotype has a specific advantage over the other two genotypes when grown in the presence of its home soil. So this red bar relative to these other two genotypes that were grown in the intact Webb Springs site. So the results suggest a novel finding that plants may evolve local adaptation to soil microbes. And as a study of performance effects, this research has applications for both agriculture and restoration ecology. Thanks.
Hi, I'm Riley Dibner. I'm a postdoc in Cynthia Weinig's research group, and I'm going to share an update of our work on how rhizosphere microbes derived from sites with differing fire histories affect the host clock. Many members of our lab contribute to this work, including graduate and undergraduate students. Soil-dwelling microbes are critical for ecosystem function. The relationships between plants and their associated microbe communities are complex. We know that plants strongly affect microbes in their rhizosphere by channeling water to the area and producing root exudates, chemicals released from the roots that include sugar and other primary and secondary metabolites. We know that a variety of plant processes and traits, such as host circadian period, can alter the composition of microorganisms in the rhizosphere. We also know that microbes feed back up, affecting defenses against disease in herbivores, access to nutrients, stress tolerance, host plant fitness, and other aspects of productivity. Recent work in our lab supports the idea that circadian period in host plants can alter microbial composition in their rhizospheres, and that in turn, rhizosphere microbes can alter clock period in their host plants. Figures A and B here show the results from an experimental study of ours of the model plant Arabidopsis thaliana. In these two figures, blue indicates a mutant genotype with a short circadian period of less than 24 hours. Orange is the wild type with a clock period of about 24 hours. And gray is a long period mutant with a clock period of more than 24 hours. Figure A contains box plots of Shannon diversity for rhizosphere microbes in this experiment. The mutant genotype with the short circadian period produced rhizosphere microbe communities that had lower Shannon diversity than those produced by either the wild-type plants or those with long circadian periods. In Figure B, the PCOA visualizes dissimilarities among rhizosphere microbes from each genotype. You can see that along that first axis, the sample is grouped by genotype. Figure C, on the right, shows the results of another clock experiment in Arabidopsis thaliana, but this time looking at how a disrupted rhizosphere microbiome affects host circadian period across a variety of genotypes. The gray boxes indicate that the host was inoculated with a sterile solution, and the blue boxes indicate that the inoculate contained an intact microbiome. For several genotypes, the circadian period was significantly longer when the microbiome was disrupted. Altogether, these results indicate that host plants and their rhizosphere microbiomes can form a complete feedback cycle on circadian period. Following these results, we have begun to investigate the presence of a similar feedback cycle in Bucca stricta, a plant native to Wyoming. We began by testing the effect of microbes in the soil on their host plants, and we took advantage of a recent wildfire to incorporate both season and disturbance severity on the host clock. We collected soils each month from April through October in 2019, sampling severely burned, moderately burned, and unburned areas. And I'll indicate here, severely burned, that's the stand replacing fire, moderately burned, that's the understory fire. We tested the effect of both types of the burn soils on circadian period in host plants, using inoculates with and without microbes. First, looking at these disrupted treatments here, these are the gray boxes in front of you. Those with no microbes, you can see that there was no effect of either season or burn, or rather month or burn. This result is what we would expect and means that the treatments without microbes have no effect on host clock by month or burn level. We did, however, find a seasonal effect with intact soils in both types of burned areas, shortening the host circadian period in August compared to April and June. The severely burned is that deeper red, and the moderately burned is a lighter orange. Those comparisons are indicated by the black asterisks here. Actually, I'm only showing the comparison with June here, but it's significant for April as well. We also observed that in the severely burned soil in August, the disrupted treatment resulted in a significantly longer host circadian period compared to the intact treatment that month. That relationship is indicated by the lighter or the blue asterisks here. We have just completed experiments to repeat these treatments across other sampling areas and including unburned soils to see if we can replicate this result. Overall, we've identified a complete feedback cycle wherein the host feeds down, possibly through the pulsing of exudates. An ongoing project in our lab is currently looking at shifts in exudate patterns over a 24-hour period. We've also found that these microbes feed back up to shift the circadian period of the host plant. We've seen evidence for this full feedback cycle in Arabidopsis thaliana and also in the native plant Bucra stricta.
Hello, my name is Brian Schumann. I'm from the Department of Geology and Geophysics. I want to speak to you today about some of the modeling efforts underway to examine and understand Wyoming's microbiome. One of the challenges that we're beginning with is the question of how do biotic communities correlate with their environment? Why is that so common in many macro communities? And is it appropriate to expect within in the microbiome? Macrobiotic communities often correlate with their environments. We see this in biomes across space, the prairie forest tundra shift that we see as you move up in elevation in Wyoming's mountains. We also see this in time, where fossil evidence suggests long-term changes in Earth's climate that are consistent over millennia to eons with what we see from other independent lines of geological evidence, such as shown in the time series here. So the question, though, is why do we see those, or why are those types of correlations common, at least at large spatial and long temporal scales? The challenge is interesting because if you imagine a landscape with multiple different taxa on it, as represented here by these different stars, squares, triangles, and circles, that landscape may be influenced by multiple different types of dynamics. On one level, we, of course, have mortality affecting individuals at each time step, say on an annual basis. And those individuals then are replaced, and they may be replaced directly by taxa that are influenced directly by, say, the climate. So different species have different climate preferences, and they then, depending on the overarching climate of the time, the prevailing climate, they may determine which taxa replace ones that have previously died. On the other hand, we also know that many communities have strong biotic interactions. So there may be facilitation or competition that directly influences which individuals are replaced. Furthermore, we also have a range of neutral processes which really are not dependent at all on the individual traits of the individual species. And so they're more or less stochastic replacement from either the local pool of possible propagules or from some regional pool of long-distance dispersal. Now, these different processes are not necessarily mutually exclusive of each other. So the modeling efforts that we've undertaken explore the effects of the interactions between these different processes to determine what types of outcomes may be realistic. So in the case I'm going to show you here, this is set up to represent realistic situations for macrobiotic communities with about 1.5% mortality per year. And each of the time series that you see here are shown in columns representing 3 different experiments. The different colored lines represent different pseudotaxa within our experiment. The model simulations are run over 10,000 years, which progress from left to right across the graphs. In each of these cases, they were forced with a climate history consistent with this realistic climate history that we've observed from fossil and geological evidence. In simulations where we include only direct climate influences, you see a progressive transition from one dominant taxon to the next as you shift through the climate space optimized by each taxon. However, many of the details of that are smoothed out, but the broad sense of one taxon dominating after another is retained even when you only have 5% of the replacement of individuals being controlled by climate, and with 95% of the dynamics controlled by neutral or often stochastic processes. On the other hand, if we replace most of the individuals according to biotic interactions, as shown on the far right here, we see that there are fundamentally only 2 taxa that really dominate over long periods of time, and that many taxa are only apparent briefly, and those are early on during a sort of successional stage where these biotic interactions determine who's first occupying the landscape. So when we look at this together, though, we can extract from this information about how well these communities correlate with the climate that was input into the model. So we create climate indices that are keyed to the climate preferences of the different taxa. And on the left here, we're showing the same data plotted over 10,000 years of those model simulations, where the gray line in the background represents the climate inputs. The black line here represents the community index associated with the climate preferences of the different taxa that resulted from the experiment in which the outcomes were directly determined by the climate preferences. And so, of course, this correlates quite closely with the gray input values overall. The orange line, however, shows how we also have a fairly strong correlation with those climate inputs, but with a modest lag when we still have 95% of the dynamics, the 95% of the replacement controlled by neutral processes. The big difference comes, however, when we look at the blue line here. The blue line represents the situation in which 95% of the interactions are controlled by biotic interactions. In that case, while there is a weak correlation overall across the whole 10,000 years with the climate inputs, there are long periods, many millennia, where there's no correlation or no significant variation in that community at all until there's periods of rapid adjustment. On the right, we see the R-squareds that represent the variance explained in these communities by the climate input data. Across the x-axis, we have each symbol on this graph represents a different climate simulation or a different community simulation. And across the x-axis is the percent of individuals that are directly influenced or controlled by the climate deterministic filtering. And in this scenario, what you can see, if you look at the y-axis being the R-squared values, is that in almost all scenarios, up down to about 5% of the individuals being replaced directly by climate effects, we retain a strong relationship with the climate. So this suggests that in macro communities, it's not surprising that we would see a broad common correlation with factors like climate or other environmental variables, because the environmental filtering strongly biases the pools of individuals that are available to interact through other dynamics, such as biotic interactions or through neutral processes. In the future steps here, we'll be modifying these experiments to look at simulations that are consistent with the microbiome and producing null distributions of outcomes that we can use to examine our data, use to understand and interpret our data from across Wyoming. So thank you.
Thanks, Brian, for talking about our efforts for Thrust 3, building mechanistic models to understand microbial ecology. What I'm going to talk about today are our advances with Thrust 3A, where we're developing predictive regression models for the distributions of microbial taxa across Wyoming. So there are a handful of challenges when trying to model microbial sequence accounts. First, the accounts exhibit high variance and a high number of zeros, where, for example, some taxon may be present and very abundant, and then not present in multiple samples, only to reappear in some moderate abundance. This variance and high number of zeros is much more than traditional statistical models can account for. And the second challenge that we have is with current sequencing technologies, we must almost always convert these counts to proportions, which creates additional challenges in modeling, such as the need for all the microbial taxa's proportions to sum to 1. And that sum to 1 constraint creates another challenge by inducing spurious correlation among the different taxa. Now, hierarchical Dirichlet multinomial modeling can address most of these challenges. The model that I'll focus on today is a regression model shown here at the bottom, where the regression model informs our estimates of the parameters in the Dirichlet distribution, the alphas, which then informs the underlying proportion of those counts in the multinomial. The Dirichlet distribution is the important part of the model that can model the wide variance seen in the microbial data, and the log link allows us to accommodate zero counts by estimating a very low underlying proportion. The next challenge we have is if we've identified thousands of microbial taxa, we will have thousands of regression models to evaluate. We cannot evaluate all the thousands and thousands of regression models without it consuming an inordinate amount of time. We need to avoid overfitting these models, such as when we have lots of environmental variables. We cannot include those predictors that do not influence the counts because that leads to overfitting. What we need are methods that can help us understand which predictor in each of the different regression models is important for all of the different taxa that we've identified. What we've been evaluating and working on and using are spike and slab priors on the regression parameters that can aid in model selection, where the prior, shown here on the right, includes a spike of probability mass at 0 to capture those predictors that are not important by driving the regression coefficients to 0. And then we have a wide slab of probability to identify and estimate those predictors that do influence microbial abundances. And so with that model, we can then learn about the inclusion probabilities for the different predictors we've measured in the environment. What's really exciting is that we can measure a few dozen or so samples across an environmental gradient, and we can then evaluate thousands of regression models within hours and gain ecological understanding of what factors in the environment are affecting microbial abundances. So we can quickly go from thousands and thousands of models that we have to often just a few dozen. That greatly speeds up our time spent on interpreting and understanding the data. Generating these sparse models is a common challenge in almost every scientific field, including biology. I am using these models to understand how climate and other environmental variables affect microbial abundances in lakes and lake sediments across Wyoming. Next, you will hear from Ella DeWolf and how we applied these models in a greenhouse experiment to understand how microbial taxa responded to a variety of secondary metabolites. Thank you.
Thanks, John. Like you said, I'm Ella DeWolf, a master's student on the project, and I'm here to talk about a project I've been working on involving the interactions of the rhizosphere microbiome and secondary metabolite production as an example of how we can use the sparse Dirichlet multinomial regression model that John just described in microbial community data analysis. First, I'd like to remind you of some of the concepts that Cynthia Weinig and Riley Dibner presented earlier with regards to plant-microbe interactions. Specifically, we know that plants feed down to affect the microbial community composition in the rhizosphere by a variety of mechanisms, including exudation of secondary metabolites. Vice versa, these rhizosphere microbes feed up to influence plant traits, including their defenses against pathogens and insect herbivores. We translated these feed up and feed down mechanisms of plant microbe interactions into research questions in the context of glucosinolates, which are secondary metabolites produced primarily in the Brassicaceae or mustard family, and contribute to insect and pathogen defense. Specifically, we asked, does glucosinolate production in the plant feed down to influence the microbial community in the rhizosphere? And vice versa, does the rhizosphere microbial community feed up to influence a plant's glucosinolate production and thereby its susceptibility to insect damage? We divided this project into 2 experiments, one to test each mechanism of interaction. I'll start with the feed down mechanism. For this experiment, we selected 12 recombinant inbred lines of Brassica rapa that, based on previous data, we predicted would have extreme glucosinolate phenotypes. Glucosinolates can be divided into several main classes based on the chemical structure of their side chain, the 2 main classes being aliphatic and indolic. As shown on the figure on the left, we chose 12 lines that would have each combination of high and low aliphatic and indolic glucosinolate levels. We planted these 12 lines in an intact soil microbiome, consisting of a sterilized potting soil and an inoculant soil to ensure that each pot started with a similar microbial community. From these plants, we harvested roots for glucosinolate analysis and rhizosphere soil for microbial community analysis. This is where the sparse Dirichlet multinomial regression modeling comes into play. While other methods commonly used to analyze microbial community data were insufficient to identify associations between glucosinolate production and the composition of the rhizosphere microbial community, this model did identify several associations. The results are shown here, where glucosinolate compounds on the left are associated with bacterial taxa on the right. Negative associations are shown in blue, while positive associations are shown in red. The strength of association is represented by the width of the line. We found that different glucosinolate compounds appear to have a different effect on microbial taxa. For example, 4-MOI3M, an indolic compound, appears to inhibit bacterial taxa, while other compounds such as 4-pentenyl and 3-butynyl and phenylethyl tend to form positive associations with microbial taxa, indicating that they may stimulate these taxa. We performed a second experiment to test feed-up interactions, that is, whether the rhizosphere microbial community influences glucosinolate production and thereby insect damage in the plant. We planted 6 of our lines in both an intact and disrupted soil microbial treatment, where the disrupted soil microbiome was inoculated with a sterilized inoculant soil to provide the same nutrients but without the microbes. We harvested leaf tissue from these plants and transferred them to the field to be subjected to flea beetles and other insects present. We then measured the resulting damage. We found that the effect of microbial inoculation on glucosinolate production depended on both the individual glucosinolate compound and the plant line in question. I show here the total glucosinolates, the sum of all of the glucosinolates we identified. We found that plant lines producing high levels of glucosinolates produced even higher levels in the intact microbiome, shown in blue, as compared to the disrupted microbiome, shown in yellow. While individual compounds showed different signals, this pattern was consistent across several compounds. However, we did not find an association of insect damage with either microbial community or glucosinolate production, possibly due to a mixture of specialist and generalist insect present in the field that may respond differently to plant defenses. To summarize all of this, we found evidence that glucosinolates play a role in both feed-up and feed-down mechanisms of plant microbe interactions. Together, these results present evidence for a full feedback cycle. The sparse Dirichlet multinomial regression model was critical in finding these associations for feed-down interactions of glucosinolates and rhizosphere microbial community. However, this model cannot tell us the causality of these associations, and future work should be done to verify the causality.
Hi there, my name is Alex Berkeley. I'm one of the co-PIs on the Microbial Ecology Collaborative Project, and I'm here to tell you a bit about Thrust 4 in our project, in which we're transforming data science capacity in Wyoming through education and outreach with specific applications to microbial ecology. Overall, our data science objective is to help researchers at all levels overcome obstacles to productive analysis of data. And I want to stress that at the beginning, that while we're enabling high performance computing and very powerful analysis in our work, on the data science side, we are also considering users at all levels in the process of doing productive analysis of data. And so we consider problems of data management and acquisition. Some of those sometimes involve the volume of the data, and so which gave rise to the label big data. But in general, data management and acquisition is a problem regardless of the size, and we try to work with students, researchers at all levels to learn best practices and methods related to data management and acquisition. By necessity, data analysis involves programming in computer code, and users at all levels benefit from advancing their skills related to programming. Also, many of the analyses that people want to do in modern science involves shared computers that allow analyses that go beyond desktop computing. And finally, one of the big challenges to being productive in analyses involves how to conceptualize our scientific questions and formalize them into some type of verbal model and then mathematical or statistical model. So overall, our data science activities relate to each of these to help people over their hurdles, their obstacles that they're facing at whatever level they are working. In terms of progress that we can report at this point in the project, we offer both semester-long and short courses and have offered them related to data science. We have a fairly active data science working group that has been ongoing. We've been engaged in a lot of consulting. A straightforward part of the project has been to invest in our shared computing system, and we've accomplished that. And we've obtained formal administrative recognition for our data science center and gotten space for it in which we can meet during normal circumstances, but not during the pandemic. The semester courses with data science components that are closely tied to this project are computational biology, which is a semester-long course that deals with fundamentals, a second course that then is a practicum in which students act as consultants for a scientific client and analyze a real-world problem. Then we have a new course that is a fundamental undergraduate course in our life science curriculum called Introduction to Research and Analysis, in which students gather data, do laboratory experiments, and analyze them all in one course and get fundamentals of laboratory science and statistics and visualization all in one. We have a long-standing and successful course in techniques in data management. We have a foundations in geographic information science and technology. And then finally, our microbial diversity and ecology course that's taught by one of our co-PIs has a significant data science component. Our data science working group is a group of graduate students, postdoctoral researchers, staff, and faculty who get together each week to pursue subjects of mutual interest. And we organize this a few weeks at a time and set a curriculum basically of what we want to learn, both through reading and discussing materials together, watching videos and discussing them, and then doing hands-on analyses. So we've read some books and done some implementation of advanced programming technologies in the last year. We've considered visualization technologies beyond standard statistical visualization that someone might encounter in the language R. We've considered D3 and various JavaScript implementations of visualization. The goal there was really to learn what exists so that we become familiar with various types of methods and different levels of visualization so that we grow our vocabulary and our understanding of what's possible rather than necessarily knowing how to do those things ourselves technically. We read most of a book and discussed a lot of concepts related to making machine learning interpretable and what types of model are particularly interpretable. And then finally, one of the really successful things that we've done, and then we'll continue to do, involve video meetings where we have a video meeting with someone who works in industry or for a government agency in some type of non-academic setting as a data scientist. And we learn both some of the technical aspects, their career path, and considerations of what it's like to work in that type of environment. We've also been able to apply our data science to the science, both of this project and to other projects. So in the video related to Thrust 2, you heard from John Calder and Ella DeWolf, where they were trying to understand how they could sort through the many possible predictors of interest and to develop sparse models. And that grew out of some of the consulting work that we do in the project between people who are more expert in data science with people who are doing the science themselves, and we're able to pull things together nicely and extend beyond what would be possible if individual researchers were trying to go it alone. and we're being able to spread some of the expertise to a broader audience. Additionally, our consulting that we do either by appointment or drop-in office hours has led to many hours each year of the project where we've consulted to science researchers from very basic questions to how do I bring my data into this software, how do I access this shared computer, to more sophisticated things where the participants actually engage in a bigger outcome like what you saw with John and Ella's work together. This year, we took that consulting to another level and went beyond the university, and students, postdoctoral researchers, and faculty consulted directly with the Wyoming Department of Health related to the coronavirus pandemic. In addition, we did some consulting for the university administration and used some of the infrastructure that we've built for collaboration within this project to share materials with various groups and to communicate. In particular, we used our online wiki that we used for sharing information and co-authoring documents, and we're able to pull University of Wyoming participants in the Data Science Center together with people from the State Department of Health to share information about models that were coming out related to the coronavirus and to do critique of them and provide critical review. So in some cases, it was us just keeping up on the literature and putting things up for the Department of Health to be able to see. And in other cases, the Department of Health was saying, hey, have you seen this? What can you tell us about this model? What's your take? And that was fairly successful. In particular, let's say late spring, going into May, the state was quite interested in a model out of the University of Washington that was influential at various governmental levels because it had very nice graphics and projections for the pandemic in different jurisdictions. And it had a large influence on policy. And there was some concern about the use of that model for projection. And so we were able to examine what the model was intended to do and how it worked, and whether it was really suited. And we concluded that it was not very well suited to project the time course of the pandemic, and was probably poorly suited to Wyoming all along through the pandemic because of the low numbers of observations that were contributing to the prior information that was coming from other jurisdictions. We were able to look at other types of prediction models and evaluate purely statistical models like the IMHE model out of the University of Washington and formal epidemiological models. And we shared that awareness with the Department of Health, and particularly a policy analyst who was really able to use us as a sounding board and make contact with various people within our data science center. We also, along the way, developed statistical visualizations that were regularly updated. This one is from May at a point in time, but that have been continued to be updated to examine geographical trends, to keep people abreast within the Department of Health and now more recently within the university administration to understand what the threat level is. This included comparing the number of deaths through time for various different jurisdictions and understanding where Wyoming fell within the overall, within the pandemic playing out across the nation. So we may have been able to help inform policy. That's hard to judge. We did write a white paper for the governor to consider. We certainly were able to build some relationships with the Department of Health and with various UW administrators, University of Wyoming administrators, with whom we had not interacted directly. And overall, it built really good experience within the Data Science Center to understand the role as consultants in an applied health context. So in terms of the data science future and where we're going to be going with Thrust 4, Overall, we've been meeting our objectives and goals quite well, and you will have seen in the annual report that the only instance of a missed goal is that we were not able to offer 4 short courses in the last academic year and in the summer. And I think part of that is can be attributed to the pandemic. But we'll address that goal again in the coming year and hopefully be able to use online resources to reach a number of people with short courses. We want to develop more working groups, including some of them off campus. We want to work on establishing our academic identity and our entity with the Data Science Center, and particularly find a place in the catalog to start listing courses and facilitate interactions between different entities on campus. We want to grow to more faculty participants beyond the group that we already have and facilitators. Actually, tomorrow, NSF EPSCoR Track 2 Award will begin at the University of Wyoming. That should help with that by hiring a large number of postdoctoral researchers in data science at the university. And I'm the principal investigator of that project, and it's a nice extension of what we've been able to do with the Data Science Center in this project. And we'll continue to reach out to other university units and companies and various partners to find common interests. Overall, we want to continue to build intellectual community and support people's shared struggle at whatever level they're at, and get encouragement from our shared successes. We, as a group of people participating in this, really benefit from the fact that we're integrating research education and capacity building. We've made good progress in the project with developing statistical analyses of compositional data, but in general, we're biologists who are statistical modelers, and so it really benefits that we can develop statistics and laboratory methods that work well together. Overall, it's provided a great opportunity for cross-training for students, postdocs, and faculty in lots of different levels of analysis that feed data into the data science effort. Thanks very much.
I'm Linda Van Diepen, an assistant professor in the Department of Ecosystem Science and Management, as well as a co-PI of the Microbial Ecology Collaborative. I'm going to introduce you to some of our great new faculty members, as well as some exciting projects that are contributing to the expansion of our research. In addition to the original science team with 8 faculty and their research group, we have expanded our capacity through the hiring of 3 new faculty members, as well as awarding seed grants to 4 different research groups. Our new faculty is represented by 2 colleges, and all 3 faculty were hired as assistant professors. In the College of Agricultural and Natural Resources, we had Woonsuk Park join the Department of Molecular Biology. In the College of Arts and Sciences, Sarah Collins joined the Department of Zoology and Physiology, and Lauren Shoemaker joined the Botany Department. As for the seed grants, we had some really great proposals submitted. 4 of them really stood out and were funded. The first seed grant is by Amy Christ's research group, and they are looking at the effects of fish introduction on the biological diversity in Wyoming alpine lakes through the sequencing of ancient environmental DNA in sediment cores. This will not only give us insight into microbial assemblages, but also other aquatic trophic levels, including the fish, aquatic and terrestrial insects, zooplankton, and phytoplankton. This will result in a comprehensive list of the taxa historically present in alpine lakes, and how diverse human influences, such as fish introductions, nutrient deposition, and climate change, impact these understudied aquatic alpine ecosystems. Another seed grant studying aquatic ecosystems is led by Sarah Collins' research group. They were looking at cyanobacterial blooms in Wyoming reservoirs, and she will discuss this in more detail in her introductory video. The seed grant awarded to Lauren Shoemaker is using protozoa and microcosm experiments to understand the assembly of novel microbial communities. And more about this exciting project will be presented in Lauren's introductory video. The last seed grant was awarded to Mike Zhu's Soil Chemistry Research Group. This project will enable us to better understand the contributions of microbes in the formation of stable soil organic matter, which can have large implications on the global carbon cycle and thus feedback to climate change. Soil samples from 3 different climate gradients will be used, including our sagebrush gradient that we sampled last summer to study the soil and plant microbiomes. The seed grant will also make use of project instrumentation housed in the Bacille lab, which includes the high-resolution Orbitrap mass spec. This will allow the characterization of the chemical composition of carbon compounds to understand the contribution of microbes versus plants to stable soil organic matter. In summary, these 4 awarded seed grants will substantially expand our research capacity by contributing to our understanding of microbial diversity and ecology in the various ecosystems across Wyoming. In addition, they're able to do some of their analysis, or actually most of their analysis, at the University of Wyoming by making use of some of our project-required instrumentation, which includes the molecular capabilities of the Genome Technologies Lab and the in-depth chemical analysis using the high-resolution Orbitrap mass spec in the Bacille lab. Next, our 3 new faculty members will introduce themselves and their research, starting with Unsu Park, followed by Sarah Collins and Lauren Shoemaker, which will also expand a little bit more about the work they are doing for the seed grants they were awarded.
Hello, I'm Insuk Park, an assistant professor in the Department of Molecular Biology. My lab started in June 2019, and currently I have 2 postdocs working in my lab, and a new graduate student will join us in this fall. My lab is interested in organelle function and dynamics in plant microbe interactions. Especially, we are studying 2 important organelles in plant microbe interaction, chloroplast and autophagosome. One major project in my lab is to understand detailed molecular mechanism of dynamic morphological changes of chloroplast and intercommunication of chloroplast and nucleus in plant immunity. When plants are undergoing their defense against pathogens, chloroplast induce tubular structure, stromule, as a protrusion from their body. Stromules provide a driving force to chloroplast to move near the nucleus. This phenotype has been well observed in plant cell under stress condition. However, we don't know the detailed mechanism of these dynamic changes, as well as what is the role of the stromule in plant defense. Currently, one postdoc in my lab is working on this project, and a new graduate student will join this project from this fall. The other main project is to use autophagy as a target to develop effective fungicide to control fungal disease in crops. We found autophagy is an effective target to control growth, development, and pathogenicity of various fungi. So we developed a convenient high-throughput chemical screening platform by using a versatile sensor to monitor autophagic activity. Using this sensor, we are screening a novel chemical which can inhibit fungi, but no effect on plant or human autophagy. Our senior research scientist and one undergraduate student are working for this project. Currently, my lab is not directly involved in current EPSCoR project. However, the resources from current project will be very useful for my research program. For example, as a part of the chloroplast morphological changes in plant immunity, we found a physiochemical status in plant cell might be directly involved in the regulation of stromule. Therefore, we are generating several sensors to monitor ROS flux, pH flux, as well as calcium concentrations. It will be very interested in screening soil microbe, which isolate by current EPSCoR project to understand their relationship with plant. This project will be helpful to optimize soil microbe, which can be beneficial to crops.
Hi everyone, my name is Sarah Collins. I'm a new faculty member here at the University of Wyoming in the Department of Zoology and Physiology, and I'm also affiliated with the program in ecology. I was hired under a search broadly defined as aquatic ecosystem ecology, and my lab works on food web linkages and biogeochemical cycles in both streams and lakes that I'll talk about in a bit. This is me over here on the right with my daughter, and the folks on the bottom of the screen right now are some really wonderful people who I've hired to work in my lab since I arrived here at the University of Wyoming. And certainly everything I'll talk about is collaborative, either with them or with other folks elsewhere, and so I'd like to acknowledge all of them in talking about this research program. So in general, my lab focuses on aquatic ecosystem ecology, as I mentioned before. I work at both local and continental scales. Traditionally, especially as a graduate student, I was trained as an experimental field ecologist. I did a lot of work in the Adirondacks in upstate New York in these forested headwater streams on the left. I also worked a lot in Trinidad and Tobago down in the Caribbean because it's a nice model system for looking at how the evolution of organisms, in this case we were working with Trinidad guppies, might interact with ecosystem processes like nutrient cycling or food web energy and nutrient transfers. So I did a lot of experimental work in the past in these sorts of systems. One is on the left here is one of my lab mates from graduate school helping me deforest the area around one of these forested headwater streams to examine how light availability influences the role of bacteria in food webs versus algal production. And on the right, you can see a setup where we're adding isotopically labeled nitrogen or carbon tracer to a stream to be able to evaluate exactly what the strength of those different food web linkages is. Here in Wyoming, I'm still doing this sort of boots on the ground field work, thinking about the role of light and nutrients in structuring ecosystems, how that influences the role of heterotrophic bacteria versus other microbes at the bottom of the food web, and in turn the kind of role and abundance of aquatic invertebrates and fish in these aquatic systems. But here in Wyoming, this is a stream from up in Grand Teton National Park. The role of light and nutrients in food webs might be quite different than these forested headwaters in the northeastern US and other locations like I mentioned before. So I'm really interested in continuing some work that we've been getting off the ground here to look at how those alpine ecosystems in Wyoming are particularly interesting for aquatic ecosystem studies. As a postdoc, I was funded through the National Science Foundation's intersections between biology and mathematical or other quantitative sciences program. And so that was really where I got my start in thinking about using large data sets to address some really similar questions to what I just mentioned, but on a much more substantial scale. So on the left here is a figure from a paper I wrote as a postdoc looking at lakes across the Midwestern and Northeastern US. So we're starting at Minnesota in the top left of these maps and headed over all the way to the Northeastern US on the right side. So these are showing concentrations of nitrogen, phosphorus, and then the stoichiometric ratio of nitrogen to phosphorus in about 10,000 lakes across the Midwestern and Northeastern United States. And so this allowed me an opportunity to collaborate with Pat Serrano at Michigan State and others who have formed a database called Logos, where all these water quality data have originated. And this was a really great opportunity because the group includes computer scientists, statisticians, and aquatic ecologists, and really taught me a lot about cross-disciplinary collaboration and analyzing large data sets. And so that's another research theme that I'm continuing to work on here at Wyoming, is thinking about aquatic ecosystem questions over large spatial and temporal scales and how we can address some similarly fundamental ideas, but using kind of large reductionist data sets rather than these difficult to execute field experiments. In particular, with the micro project, I have a new seed grant. It's about harmful cyanobacterial blooms in Wyoming reservoirs. So this is a project that I've been working on with funding from our state water development program. But that's a particularly applied funding source where we're out sampling and doing some nutrient manipulation experiments to try to inform policy and management. The seed grant is allowing us to try to use genomic methods from the micro project to ID the species composition of harmful algal blooms, so which cyanobacterium in particular is responsible for a bloom, and try to relate that to toxicity, other underlying conditions, and just kind of the microscope count data for what cyanobacteria are present compared to these genomic methods. It's also allowing us to do some lab experiments in bottles where we're manipulating light and nutrient levels and the form of nutrients available to try to figure out how cyanobacterial blooms might form in response to those drivers. So my lab technician, Jade, has been working on this project this summer so far. We're just starting to get samples in from our collaborators with the State Department of Environmental Quality and extracting DNA, and she's been hard at work there. And I have a new graduate student who will be starting on this project next month, so we're excited to get that off the ground. So with that, I'll sign off for now, but thank you for your time and attention.
Hello, my name is Lauren Shoemaker, and I joined the University of Wyoming as part of the EPSCoR faculty cluster hire, joining as a computational biologist in the botany department. I began as an assistant professor in fall of 2019. Most broadly, my research combines theory and modeling to look at population and community ecology, examining patterns and species abundances across space and through time, and then scaling these patterns up to overall community dynamics, thinking about species coexistence and community richness. Before beginning at the University of Wyoming, I did my PhD at the University of Colorado Boulder in ecology and evolutionary biology, along with a certificate in interdisciplinary quantitative biology. From there, I received a James S. McDonnell Foundation postdoc in complex system science, which I took to the University of Minnesota. In my research, I combine theoretical models with empirical tests as well as synthesis. So theory and models of community dynamics are central to most everything I do. And then I take the predictions from those theoretical models and examine how we can test those predictions empirically, whether that's, say, using microbial microcosm systems or field experiments, and then from there, seeing if we can scale those up even further to more global synthesis measures. Currently in my lab, we apply that approach to focus on a couple key questions. The first is examining how abiotic variation in the underlying environment actually can alter the strength and potentially even the sign of species interactions, and the ramifications of this for long-term coexistence and species diversity. And again, this is in thinking about microbial systems, but then also even thinking quite broadly, say, for example, food web dynamics in the rocky intertidal. Second, I've been working a lot recently on how scale dependency can help, or in some cases hinder, our understanding of what mechanisms drive key patterns and connecting mechanisms to these patterns, specifically in a scale-dependent way. I was also one of the recipients of the EPSCoR seed grant for a project looking at assembly of novel microbial communities. And this is in collaboration with Jeanette Davidson, who's a master's student in my lab. And through this seed grant funding, we've been able to support Jeanette on an RA, as well as Annalise Brunner, who's an undergrad in microbial biology. So for our seed grant project, we're really taking these community assembly ideas and connecting them to invasion ecology, examining how the mechanisms that determine how a community assembles has ramifications for its resistance as well as resilience to the invasion of novel species. And we're taking, again, this multi-pronged approach. So we're actually adapting theory that was originally developed for plant communities and applying them and extending it for microbial community assembly. And then testing this theory using a microcosm system consisting of several protist species, where we vary environmental conditions as well as then different species combinations, and therefore the strength of competition between species. And the eventual goal is that this will provide insight into community assembly and invasion dynamics for both microbial systems, as well as then taking this and extending it back to these plant communities and thinking about it in macro systems as well.